All right, so we're about to, did we finish the vessels chapter? No, I didn't think so. Okay. So we're about to really finish this up, though. So we're going to finish this up in the next, actually, few minutes, probably. And I want to let you all know that, you know, you have a test coming up on the cardiovascular system, which is hematology, the heart, and then what we got out of these vessels chapter, right? Did y'all already decide when you wanted that?
It's Thursday.
So you did. Okay, so good. Everybody's on board with that, right? So Thursday morning at 9 o'clock, we'll have that. When y'all have that, you're going to also label the heart. Megan, tell me, where'd you let that make? Come on with it. It said that I forgot, right? But what y'all have decided. But that's fine. So I want you to also know you're going to label the heart. Let me just briefly again list the structures of the heart that you would be responsible for. You may not have a word bank, but you would have something that would denote that you need to label this. You know, there might be a lot of lines, but I would have lines denoted that you would have to label, okay? So it might not be a word bank, so should you be looking at spelling?
Yes.
All right, so you want to know the pericardium, the epicardium, the myocardium, and the endocardium. What are those all four related to? It's the sac is the pericardium, the epi is the outer layer of the heart, the myo is the muscle layer, you know, thick muscle, and the endocardium is where? In the chambers. It's lining the chambers, isn't it? You think you need to know the four chambers? Four chambers. Right atrium, left atrium, right ventricle, left ventricle. How about the four valves? You need to look at a ton of pictures so you know where those valves are. Tricuspid, bicuspid, also known as mitral. And then the pulmonary valve and the aortic valve. You're going to know those? Okay. How about some vessels leading to and from the heart? How about inferior and superior vena cava? I think you would need to know that. How about the pulmonary veins, the right and the left pulmonary veins, right? Correct? Okay. And then how about the pulmonary arteries, right and left pulmonary arteries that come off that trunk, that pulmonary trunk, right? And how about the aorta? You fair enough? And then also, I talked to you guys about the fossa ovalis. I want you to look that up. It's like a little, it almost looks like a dimple between, in that wall that separates the right and left atrium. It almost looks like a dimple. What was it at one point? It really was a hole, wasn't it? Yeah, okay. So guys, could y'all have that today? You want to take that today? All right. But you'll do it Thursday, right? It'll be good Thursday. So you'll do that first at 9 o'clock, and then you'll have a 50-question multiple choice test on the cardiovascular system. You think in hematology you'll need to know all the functions for all the different cells? You do? Okay, so you will. That's true. So you're going to need to know that. You know, and some of the disorders that we talked about, like sickle cell disease and maybe, you know, polycythemia and the different types of anemias. Are there more than one type of cause for anemia? Is there more than one cause? Yeah. Okay. And then what are leukemias? Cancer of the white blood cell line, right? So leukocytes or white cells, but leukemias are going to be cancer of the white cell lines. And then you would need to know which white cell line, but you don't need to know this for the test. You just need to know that's what leukemia means. So you're going to make sure you go back and review all the lecture notes that we had. Maybe study the study guide to look at, so you and your classmates have kind of covered the bases there. And do you have any questions for me about that? Are you good? All right. Well, let me tell you again just to remind you about scheduling to come. We're going to finish this vascular thing today, and I'm going to actually assign for you. It will not be Thursday. It will be next week, but I'm going to assign for you another lab opportunity. We're going to call them an opportunity. And next week in lab, during that Tuesday in lab, the opportunity you're going to have is to label the major systemic arteries and the major systemic veins, because that's about really all we have left to talk about in this chapter that we'd already talked about. Some other stuff. I'm going to get back to some of this, but anyway. Okay, it's a picture of him somewhere. Here they are. Look at this, major systemic arteries. Arteries carry blood away from the heart. And I know you look at this list and you think she's lost her friggin' mind. No, I haven't. Because you all know these areas because you know the bone. What is this bone? The radius. What's this bone? So what is this artery? The radial artery. What is this artery? You get what I mean? How about here? What's this bone? The femur. So what's this major artery? Femoral. So you get what I mean? What's this region behind the knee? Popliteal. Some people say popliteal is another. How do you say it? How did you learn to say it? Popliteal. So anyway, whatever. Don't have to spell it, right? So, but y'all learned regions, didn't you? You learned that brachial is always referring to the what? The arm. So these are not as hard. Axillary, y'all knew was what? Pits. It's not just that. Okay, so armpit. So this is not as hard as you think. I am telling you right now, so it's fair. And it's on recording too. It should be. There's one they left out on this. Now look, this is going to, I'm going to warn you, this is different from the one in your book. So just to be fair, since people have different editions of the book, I'm going to use this list that's on the PowerPoint because you all have access to this PowerPoint through Canvas. But they left one out they shouldn't have, and these are the carotids. And y'all all know the carotid arteries are in the neck, don't you? So you already knew that, but make sure you add it to this, carotids. Now, we know we don't just have arteries. We know these arteries are supplying blood to oxygenated blood to our system, our body's tissues. But we know that blood is then moving into vessels, the capillary vessels, and then into what? Veins, visuals and veins. So we know actually on top of all this is a whole nother set of vessels, don't we? And guess what I just told you what they're called. What are they called? A major systemic what? Veins. And I want to bring your attention. To this slide, and this slide is 67. These are the major systemic veins that are returning blood to which pump? The right pump, right? The right pump, the pulmonary pump, because it's going to then send it back to the lungs. So y'all just did that, this red blood cell. So these major systemic veins are a lot of these words sounding the same as what we just learned on the other, but these are the veins. Now I want to add something here because they didn't, I don't know why they don't have it, but the veins that are bringing blood back from the head are called jugulars. Okay, jugular veins. So add that, and will this be a fair kind of lab quiz for next Tuesday, a week from today, right? Because y'all will look over this, make sure that you could label these. Knowing the bones and knowing the regions should make this kind of elementary. But just in case there's something on here that isn't new to you, or that is new to you, make sure you look over and you know what these are, okay? So this will be a lab quiz next Tuesday, fair? All right. Now, great. It's just a couple more things I want to do on this chapter, and we'll be done with this chapter. Now, I do want you to realize that I didn't cover everything in here. If I didn't talk about it, will you have to know it? Unless it was in web. Deeper insights or disorders tables. So some of this, you know, I didn't, so it's FYI. But if I talked about it, we know it could be a fair thing on your test, okay? So that will be, we'll finish that up in just a second. This set of information. We're going to then start, I want people to, because we only have about 10 minutes left in this material. And then we're going to start on the lymphatic system. So we're going to start a new set of information. But the lymphatic system and the respiratory system, 2 chapters that we would probably get through probably by the end of this week. I think that's going to be y'all's take-home. I'm going to let you do that as a take-home test. And because next Thursday, do y'all remember that next Thursday I'll be out of town for a conference? I had told you all that, hadn't I? No. I hadn't? No. Really?
That's okay.
That's perfectly fine.
That really worries me, and I didn't tell you all that.
The 11th.
Okay, so yeah, that's the 11th, and I am going to be out at a conference in Roanoke. So I'm going to be gone on a Wednesday, Thursday, and Friday. I will still be available by email if y'all have any questions. But what I think I'm going to do on Monday is give you all that take home at the end of Monday. You know, I need Tuesday. That's what I meant. At the end of Tuesday, at the end of that class, I'm going to give you all that take home, open book, open notes on the lymphatic and respiratory system that we're going to start talking about today and finish by Tuesday. We meet on Tuesday, Thursdays, right?
Yeah.
Yeah, Tuesday. So anyway, so that you all, that day that I have to be out, and y'all don't need to show up on that Thursday, the 11th, right? You all would have something that could keep you occupied. Not that I mean to keep you occupied. I know y'all wish, but you get what I mean. And this is going to be an open book, open notes type of test. It will be exactly the same you would have had in here proctored. So don't think I'm going to be one of those people who give you an open book test and you look at it and think, where the hell did this come from? It's not. It's going to be from where? Notes that we go over, so still continue to take notes because you'll be able to find those answers right away. There's one rule on this open book, open notes test. You may not ask another living soul for help. What does that just mean simply? I just want you to do your own work, right? I want you to do your own work. Don't be calling up and saying, I can't find that anywhere. Can you help me with this? Right? Jessica, you good?
Yeah. I was adding it to my
Okay,
calendar.
okay. So, so is everybody good with that? I think that gives us a reader, and I think y'all deserve it because there's something I'm going to ask you all to do, and I'm going to give you credit for this too. I actually got my hand for this. Y'all don't care if I got my hand. But your class got chosen to do a survey, and I'm jealous of y'all. I don't want to give people your time. Unless it's after our time. It needs to be our time. But this is something that, okay, I get that they need this information. So it's a survey, and they say it takes, I don't know, I don't think it's going to take 30 minutes, but it's not a survey for this class, by the way. It's not. It's something it's a national thing for this community college. But anyway, and I'm going to give you all, I don't care, y'all can decide if it's a lab participation grade quiz or if it's a lecture. I guess lab would take better. So, and we'll do it during lab time. So anyway, we'll do that. And I feel like y'all need to get credit for that. They randomly, they say, choose classes, and then we're required to administer it. So I'm required to administer it to y'all. You're the class that I chose. You're one of the classes. Did anybody take this in another class? By the way, this is called assessment survey. Did anybody take this in another class? Okay, so gosh, really unlucky of you if in every class you chose classes that got picked for the survey thing. It has nothing to do with anatomy and physiology. It won't have anything to do with this class, but it's something I'm supposed to administer. But I'll give you all credit for doing it because I think you deserve it. If it takes your time, time's valuable. All right, so we'll do that maybe at the end of today or at the beginning of the lab or something. Okay, let's finish up. Y'all have any questions for me about what's coming up in the next 2 weeks? So you've got a test Thursday, and then you have the opportunity for an open book, open notes test on the lymphatic and respiratory system that we should finish up the notes at least by next Tuesday, and I'll give it to you so that you have it during that Thursday that you don't have to come in for me. Does anybody have any other classes on Thursdays that you usually are waiting around here for to go to? So it saves y'all the gas to come in. You don't have to come in here, right? Okay. All right, so let's go ahead and finish up this though, and then we'll start lymphatic. We talked about shock. What's the most common type of shock? Hypovolemic shock. Hypovolemic shock. Most common kind of shock. And this is going to be because of dehydration, burns, bleeding that can occur because of trauma or could occur because of pathology. Now, obviously, trauma, we can have bleeding, but we can also have bleeding because of like gastric ulcers, or just there are many different blood conditions that can cause bleeding. So this is hypovolemic. So low blood volume, right? That's what that would literally mean, that word. Now, we can also have tumors or aneurysms. So an aneurysm, he says, if they rupture, of course, you're going to be losing blood volume. So these are other reasons that you can have that. You can have vascular pooling or venous pooling and give you vascular shock. This is why they tell you if you're standing up for long periods of time, don't lock the knees, right? Because you see people like that will pass out with this. And that can actually cause people a lot of damage if they fall and hit something, hit their head, or knock their teeth out, or whatever that can happen. Usually, you're going to compensate for that as soon as you get horizontal. As soon as the person's horizontal, they may be knocked out and maybe knocked out their teeth. But usually they'll come back too after that because the blood will keep circulating. Septic shock is because of an infectious organism that is actually causing this, where the sometimes the cells will be lysine, or the capillaries just get leakier because of some of the toxins that are being produced. So this is called septic shock. If y'all have ever heard of sepsis, sepsis means actively growing microorganisms in the bloodstream. Actively growing microorganisms in the bloodstream. This is a leading cause of death in people. And sometimes it's not actually even just put on the death certificate, it's just assumed people knew this. So somebody might say that they died from cancer, but a lot of times it's that the cancer in end stages makes their immune system so weak that they end up getting an infection, whether it's urinary tract infection that goes to the bloodstream, or whatever infection it might be. Our dean of nursing I didn't realize what she died from. Well, she didn't die. Her mom died. But anyway, her mother passed away about a week ago, and I saw our dean of nursing yesterday, and I didn't realize why her mother had passed until she told me yesterday that she had influenza B, and it had gone into complications, and she became septic. And it's serious, isn't it? Even people with the best treatment that have sepsis, up to half of them will die. So this is like a no joke thing, have actively growing microorganisms in the bloodstream. So anaphylactic shock is when you have severe hypersensitivities. So y'all have heard of severe hypersensitivities where lots of histamine is being released, and bee stings, peanut allergies, shellfish, what else? Some antibiotics, bees, so bee stings. So this can actually, these are serious situations that can take someone into shock. So usually if you compensate, then the person's going to, if the body's going to get back in homeostasis. But if it's decompensated shock, then without medical intervention, then the person's going to die. Because cardiac output's going to go so low. What did I tell you? If your blood's not pumping fast enough around, what will the heart get a signal to do? Stop. So you'll go into cardiac arrest. So ischemic attacks mean lack of blood supply, lack of circulation. That's what that term means, so you should know that. And if you're not getting enough circulation to the brainstem and to the nervous system, then the nervous system controls all other homeostasis, doesn't it? So hopefully that kind of makes sense to you. I need you all knowing exactly what these two abbreviations stand for. TIA stands for, you're going to be asked this on the test. Is that like a gimme questions, right? TIA stands for transient ischemic attacks. Transient means it comes and goes. It doesn't stay, it just kind of comes and goes. Transient, moving, right? So ischemic means there's going to be a lack of blood supply. These used to be called mini strokes or warning strokes. Now they're considered strokes. They are considered strokes. The difference in a TIA and a CVA, cerebral vascular accident, which is a stroke, is that the signs and symptoms of a TIA will usually go away within a relatively short period of time, 24, 48 hours, 72 hours. So you might have initially some paralysis, some speech impediment. You might have initially some weakness on the side, but usually those symptoms kind of go away. Some people don't even, acknowledge that they've had that until they've had a CVA and survived the cerebral vascular accident. Some people don't survive it, but the people who survive it will then, if they can communicate with you, will tell you You know, over the last couple of years, I've had these periods where I've had weakness on one side, or I've had these periods of time where I felt a little numb. All right? But what were they really having? They were having TIAs at that time, which are strokes. Okay, so it's really it's something to not ignore, right? What is the FAST thing? What do they call? What do they mean when they talk about the FAST? If you suspect someone's having a stroke, you're supposed to remember FAST. So F is face, A arm, S speech. Yeah. So the face is like smiling or whatever, you know. And then the S is speech, heavy talk. And what was T then? Time. I really had to ask you. I forgot. So so that's kind of what you're supposed to be thinking about if you suspect somebody's had a TIA. All right. So good. How y'all feeling about the cardiovascular system? Some of this other stuff we do in our next information. So so I don't hit it here. I'm going to hit it in the basic organ systems. How are y'all feeling about heart, blood, and vessels? Okay. You're feeling like okay? Got notes on that? I know what you've done. All right. All right. So guys, this information, and by the way, I understand that you're not going to have a lecture quiz Thursday. You're not going to have a lecture quiz because you already have a lab quiz and a test set up. So this lymphatic information we're going to start with today, and we'll finish with and do respiratory on Thursday. You're not going to have lecture quiz. You won't have lecture quiz on this, okay? I know you're going to be taking good notes, though, so that when you do get that paper test, it'll be rather simple for you to kind of go through and get this down. So is everybody taking a deep breath and relaxing over this? All right. All right. Okay, so let's talk about the lymphatic system. I want you all to tell me how much you know about the lymphatic system. So when you think about, call this our lymphatic man. You know, we just saw the major arteries, didn't we? And we know we have these major arteries. You can feel pulses, right? Kind of pulses. You know, you can feel the pulses. Carotids. You're not supposed to feel those too much. So anyway, but we know we have veins. So we know we have these vascular veins and stuff. And we looked at the arteries that we know are carrying oxygenated blood in the system and the system, and the veins that are carrying deoxygenated blood back to the right side heart in the system. And we know that the larger ones have a muscle layer wall, right? Because the muscles are smooth muscles that can contract or dilate, right? And so you know there's significant vessels. Did you know you have another entire vascular system though? Did you know you have this lymphatic vascular system? It's really not green, but they made it look green on here, right? But anyway, so what is it transporting?
Fluid.
What kind of fluid?
Tissue fluid.
Tissue fluid? Tissue fluid, so what do you mean? All right, so really we're trillions of cells, is that right? We're trillions of cells that aren't flat like our pictures. Our cells are three-dimensional, right? And what are our cells filled with? Well, they're protein factories, but they're filled with intracellular fluid. The cytoplasm, we learned it as the cytoplasm, it's intracellular fluid. Cells are filled with mostly water. And cells are bathed in mostly water. They're bathed. Even the cells that are really close together, there's a fluid interface. And so cells are kind of bathed in water. Cells are filled with water. And do we know how water moves between two compartments? What did we call the movement of water between two areas of concentration? We called it what? Osmosis. We called it osmosis. And we know water is just sort of freely moving. Water will move down its gradient, though. You remember that? So if you got a higher concentration of water here than you do here, and the membrane is separating, water will move in on it until water is balanced between the two. But even when it's balanced between the two, water is still kind of moving in and out, right? You with me on this? What was the term osmolarity? What did that refer to? Did y'all hear all that? That's right. It means osmolarity means that you know how many solutes are in that solution. Certainly there's a lot of water, but you know how many solutes, and the percentage of solutes in each area is referred to as the osmolarity. So the osmolarity of the extracellular fluid and the intracellular fluid had better be kind of balanced, because then that would mean that the water's just kind of moving in and out, moving, always moving, because stagnant water's gross. Who's ever seen stagnant water? Unless it's mud hole, then it's fine. But no, pretty much stagnant water is gross. Okay, so but water isn't stagnant in your body, it's constantly moving in and out and in and out. And the osmolarity of the fluids, the extracellular fluid and intracellular fluid, and your blood plasma play a role in how water's moving. Would that all make good sense to you? If I told you that your cells, I don't have a picture of it, but if I told you that your cells, like let's think about your cells that are just like any cells, I don't care, pick an organ, are being bathed in fluid that's mostly water. Where is that fluid coming from? Because you said it's water is always kind of moving, because it would be nasty if it wasn't. Nasty is probably not a really professional word, but okay. But you get what I mean. Water's kind of moving in and out. So solutes are diffusing, you know, and sometimes actively and sometimes passively. But there's this constant movement and motion. But where does the water that's bathing our cells and interchanging in our cells to, where's it all coming from? Where's that water coming from? What'd you say?
No.
Where, no. What's lymphatic system? It's coming from the fluid that you take in, you drink, right? It's going to your bloodstream. It's going to your bloodstream. And then as this blood gets taken to capillaries, what did I tell you happens at the capillary level? I said exchange of nutrients and waste. What's definitely a nutrient? And what can be a waste? Water, right? Right. So, and where is water found in blood? What part of blood is water? Plasma. And what component of plasma is water? Is it the predominant component of plasma? Yes. It is, way far away. The predominant component of plasma is water. So in the capillary level, which all of our tissues have capillaries feeding them, all of our organs have capillaries feeding them, water will move out into the tissue spaces. Let's think of this as being our little toes, our pinky toes, okay? So water is moving out. But wait a minute, there's already water there. So what is that putting a little bit of pressure on? The water that's there, right? The water that's there. And so the water that's there is going to move into these closed-end capillaries called lymphatic capillaries. Lymphatic capillaries are different from blood capillaries because blood capillaries are closed. It's a closed system, right? It's a closed system. From the left side of the heart going back to the right side of the heart, closed system. But water and nutrients are moving out and waste are moving back into blood. But as water moves out, it's moving the water that's bathing our cells, because that's bathing suggests that it's rinsing, right? Rinsing. And as the pressure gets put on this fluid, it moves into these closed-in capillaries. They've got closed ends. So that as water moves in these, it has to go in one direction. Does that make sense to you? Now I'm going to get back to another picture. It's not to let me.
So when there's too much of
Wait a minute. Can I just say one other thing, show you one other picture? Just so you can see right here. So let's do this. I'll be sad I did this because I'm going to be still. So if we look at our blood capillaries, so water's moving out, bathing our cells, but the water that's already here is going to end up moving into these little lymphatic capillaries. And it won't be able to leak back out because the ends of these are closed. These are microscopic vessels. And then this fluid is going to move through these lymphatic capillaries and be pushed in kind of one direction. Okay? Now, the fluid that is bathing our cells, guess what we call it? Lymph. We call it lymph. So we call this lymph. And Andrea, ask me again. What you
This picture was a lot better. I was gonna ask, basically, is the extra cellular fluid what gets soaked up in there, if you will, like goes into that.
Yes, the cellular fluid that's there is being pushed so that you're getting this constant refreshing of fluids, which we said even sounds like a good idea, doesn't it? Okay, so this constant. So what lymph really is, we said it's coming from blood, the water coming from blood plasma. So if I were to ask you where does lymph come from, you're going to tell me it comes from blood plasma, right? And lymph is actually water that's very similar to blood plasma because it came from there, but much less of the proteins that we think about that we find in blood plasma, much less of those, but it's still very similar to it. It's really, you know what lymph really is? It's the fluid that's bathing our cells. And lymph is then going to feed into these lymphatic capillaries. And then these lymphatic capillaries, and I need y'all to kind of picture this. These, and you can see an actual picture of a lymphatic vessel, because they really aren't green, obviously. From these capillaries, now I'm not going to ask you to know this anatomy, but would it make sense to you that these capillaries, which are microscopic, just like our blood capillaries are, you know, one cell layer thick, they're so tiny that red cells have to move through single file, right? That maybe it makes sense to you that they enter into slightly larger and less numerous vessels until they're in pretty big vessels at this point, so pretty big vessels, lymphatic vessels, until they actually empty these lymphatic vessels back to the bloodstream. So lymph comes from blood plasma. It's nourishing, it's bathing and replenishing and cleansing and keeping chemical stability of our tissues. And then it's eventually getting returned back to where? The bloodstream. Now I want you to look at this guy here, lymphatic man, or we could change him into a woman, but whatever, we have these lymphatic vessels. And I want you to notice that every now and then, if you look, you'll see these little swollen looking areas. And what these are nodes, lymph nodes. So even in this illustration, it looks like lymph has to move through these little nodes, has to move through this node before it's allowed to continue on. Is that right? And so the function of a lymph node is the place where actually lymph gets filtered. Are you imagining your little toe? Are you feeling your little toe at this point? Everybody got two little toes, right? You feeling your toes? How many times have you ever thought about feeling your toes? Whatever. But is everybody sort of in tune with your little toes at this point? All right, so I want you to think about this. I really want you to think about this and realize that the fluid, lymph, the bathing those, has to move through these kinds of vessels, coming all the way back up through, and it will go through these nodes. These nodes are going to actually contain a lot of white blood cells. They're going to have, it's going to move through these little ducts in the nodes that contain white blood cells. And right now, I want you to think about stumping your little toe. You stumped it so hard that it's bleeding. How many of you have ever done that? You were sure you broke your little toe. It's bleeding. It's stumped it so hard. But if you stomped your little toe or you stepped on something, and now you can introduce microorganisms into the tissue, right? Because do we live in a sterile world? No, we don't. And so the skin's broken, whatever you did to your little toe, if the skin's broken, the tissue fluid that's bathing it, that's okay, but that fluid's going to be moving through this lymph, and that lymph has to go through these nodes. Don't you think it's a great idea that it can be cleaned before it's allowed to pass through here? So there's tons of white blood cells in here that will detect anything that's foreign and absolutely, hopefully, take them out right here. So as the lymph is allowed to progress on its way, it's hopefully going to be cleared before it has to get back all the way to where? To the bloodstream. Does it make sense to you that if, because they're just seeing an illustration here with one node and one node, but does it make sense to you that lymph usually has to move through quite a few different nodes? Because it might not get clean with the first one, but by the time it's going through the second, third, fourth one, hopefully it's gotten filtered. So the function of a lymph node is to do what?
Filter, like, blood.
No, filter the lymph. Why is it not filtered?
Infection, does that mean that nodes are not working?
So, how many of you have had swollen lymph nodes? If you haven't all had swollen lymph nodes, you probably not lived and not go out and play in the woods or something. But actually, so swollen lymph nodes means that the nodes are working, Jessica. They're working. So that's not a bad thing. That's actually a good thing because it means that the nodes are actually doing their job. Now, can it be uncomfortable for you if you have swollen lymph nodes? And can it be an indicator as a sign to your practitioner that your body's fighting something? Yeah, it's a really clear sign that your body's fighting something when somebody has swollen nodes. How many of you have felt swollen nodes even behind the ears and in your throat, maybe even in the inguinal region, groin region? Now, does it make sense to you that around the areas where we have portals of entry, you may not have really ever thought of your mouth and your nose and your eyes and your ears as portals of entry. But as an infectious disease person, I think of your openings in your body, whether it's vaginal, urethral, rectal, whatever it might be, as portals of entry. That's what I think of them as, because they really are portals of entry, aren't they? And this is how a lot of things enter into our system that can actually cause disease. And does it make sense to you that you have a ton of nodes in these regions? If anybody walked by and I was doing this. But does it make sense that you have a lot of nodes in those regions? Does it make sense that because we walk on our feet, meaning we can hurt our feet, that we have a lot of nodes along the way to get back to our system? We have a lot of nodes behind our knees. We have nodes here. We have nodes under our arms because we also work with our what? Our hands. So we have injuries to our hands, right? All right. Did I answer the question? Are you sure? Yeah. Okay, so I forgot the question. Okay, so I want to show you this because y'all have all heard of people who have cancers, whether it's breast cancer or wherever it may be. You'll hear about people who have cancers that do have surgeries. Not all cancers are surgery. You're able to do surgery, but the ones that do, that they'll remove some nodes. Have you heard of that? All right, and you've also remembered that different types of tumors, you can have benign or malignant. And so we said the difference between benign and malignant tumors, if you remember from AMP1, is that a benign tumor is encapsulated, meaning all the tumor cells are staying in place. They may be getting too big and they may be compressing vital organs, but they're staying in one place. But a malignant tumor is not encapsulated. And what can happen is if there's a malignant mass growing, cells can break loose. Cancer cells can break loose. And we said that what kills people who have malignant cancer is really not the local area where it started, it's that the cancer has what? And what is metastasized? So they have metastatic cancer. So how does cancer spread from the site of origin? It spreads usually, we know, through what system? The lymphatic system. So what they do is when they go in and remove, like maybe a lumpectomy, they will actually, when they're taking that tissue with a margin around it, they will right there put dye in the tissue, and they watch the dye move through, clear from the tissue space. The first node that the dye gets to is called the sentinel or guardian node, and they will remove it. Then they will remove usually a certain number past that point, depending on the surgeon, what they've decided. Now, what we like to see is when they do the pathology on their node, those nodes, what do we like to see? What'd you say? You like, does everybody getting that when they send those to pathology, and so pathology is going to freeze them and do histology slides, and they're going to look to see in those nodes if there are any cancer cells. What we like to see is that there's no cancer cells found in any of the nodes. So that's usually referred to as no node involvement, because then it's less likely that a cancer cell has gotten through got into the bloodstream and then jumped off somewhere, like the liver or the brain or bone. You get what I'm saying? So, is it a sure bet that it hasn't? If you have no node involvement? No, but it's a better prognosis if there's no node involvement, or if the percentage of nodes that were positive was really low. The more nodes that were involved, the more likely that it's gotten through. Some cells have gotten through. Any questions about that?
What was that called, guardian?
Guardian, like the guard, you know, a guardian or sentinel, like a guard or sentinel of that region. So that's just referring to the first nodes that are clearing that area, that are protecting that area. I'm not going to ask you that, but you will hear about that. So I don't think I have any questions about it. But aren't y'all interested in that? I mean, that's what they do. That's what they're doing, no matter where the cancer is. If they've surgically gone in to remove it, they're going to remove some nodes. Now, let me say this. Suppose they did a whole radical mastectomy. I mean, even a lumpectomy, they're removing some margin tissue in some of these nodes. So what will it mean for tissue? And I want y'all to think about this. What will it mean for tissue that has lost that lymphatic return? Because this is the fluid that's bathing here, and it needs to get back here, right? So what happens if that's going to be compromised? What would happen to those tissues? Swelling in those tissues. You can have excess swelling in those tissues. It's more difficult for those tissues to clear. So you're going to be really careful with somebody if they've had any kind of surgery on a particular side, you know, that you are not going to put tourniquets on, like blood pressure cuffs, are you? Or tourniquets to draw blood or to start IVs. You're not going to use that side, are you? Right? Because they already have impairment. And that's why you want those stigmomanometers and even tourniquets for phlebotomy. Those are tourniquets, right? So they're impeding blood flow and lymphatic return. And so you're not going to do that to people who have had those on that side for the rest of their life. Sorry. Right. Right. Now, there are things that they can do if somebody has had lymphatic damage for whatever reasons. There are things that they can do. Can they use gravity to help them? Can they use gravity to help with the lymphatic return? They can. Can they use massage and physical therapy sometimes for that? Yeah, they can. They can use that for that and try to help with that. So there's definitely things they can do for lymphatic return. Any questions for me? So you wouldn't be able to tell me what lymph is. You wouldn't know where lymph comes from and where it ends up going back to. And also the function of a lymph node. Is that fair enough? Mm-hmm. Function of lymph nodes. Now, do you think the pressure on lymph is great? Do you think that the pressure from your little toe, that fluid that has to get all the way back up here to the subclavian vein, do you think that the pressure is great on that little toe? I don't feel much pressure on my little toes, y'all. Except for overweight pressure, but no, other than that, I don't feel pressure from the lift, okay? And it's not. It's really low. It's so low. It's kind of low like venous return is low. So we said in veins, we have something that we don't need in arteries. So what do you think we have in lymphatic vessels that are in veins too to help with one-way flow? Valves. And they really do. But what's another thing? The best thing you can do to help with lymphatic flow is just to keep what? Is to keep moving. And it's so important that we keep moving, not just for our right venous return of blood, but also for the return of lymph. Because that's contributing to blood volume. And that's helping again with our right venous return because it's stabilizing our blood volume. So go figure, we're supposed to always be moving, right? Good? All right, so hopefully that makes sense. Even when we are walking, whatever, if this were a vessel, if this were a lymphatic vessel or a blood vein in the lower leg, when your calf muscle is walking, even just walking, it's actually milking that to keep it going up, right? So just walking. But even when you're breathing, what's that dome-shaped muscle when you're breathing that's changing shape? So even when you're breathing, you're changing the thoracic and abdominal shape, and that's even helping those vessels that are in those areas, those lymphatic vessels, that's helping keeping it going back in the one way that it's supposed to be going. Exercise help? I mean, even increased exercise, not just walking, that can help too, right? So I hope you realize that. I like this little illustration of this lymph node because it kind of makes engineering sense. So you've got these very small little vessels bringing lymph here, and it kind of backs up in the node as it's designed to do because there's only like this one exit. Do you see this? So you've got a lot of little ones bringing lymph, but it kind of backs up, and that's okay that it backs up because if we blow this little area up here, we see that we've got a lot of what? What is a macrophage? It's a big eater, which is derived from white blood cells, monocytes, right? And how about these? Do y'all know about lymphocytes? You're going to know more about them in a little bit. So lymphocytes. So we've got this beautiful kind of system that is clearing our lymph before it's allowed to exit. But even if something gets by that one, might the next node catch it? So swollen lymph nodes are actually a sign that your body's lymph nodes are working. Now, can they get swollen? Can they get weak? Yes. They can, and it can actually take a long time for lymph nodes to go down. It can normally take weeks for them to go down. So some people are kind of freaked out by that because they see swollen lymph nodes and they immediately think what Hodgkin's or something. You know, they think lymphoma. But and sometimes that's the case, or not. You know, sometimes it is, but not usually. Usually it's just that your lymph nodes are doing the job that they're supposed to be doing. You know, so it's not a bad thing. And it can take a while for them to go down. All right. Now, tonsils. Tonsils good or bad? And we'll take a quick break. Tonsils good or bad? Huh? Yeah, you know, so tonsils are good. Tonsils are good. How many of you had bad ones? How many of you had your tonsils out? Raise your hands high if you've had your tonsils out. Only 2 people in this room have had their tonsils out. I'm proud of that. I'm happy about that. How many of you should have had your tonsils out? Okay. Yeah. So anyway, well, look, it's a fine line. And some people, ear, nose, and throat, ENT stands for ear, nose, and throat specialist. But, you know, at one point they removed everybody's tonsils. I don't care what, they were yanking tonsils. But we realized tonsils have a really important function. And you have different ones. And I would want you to know the names of your tonsils and their locations. So I would want you to know this. And of course, there's no picture because that would just be too convenient. Hey guys, when somebody says, and you see those tonsils on the sides, everybody thinks of your tonsils as being those, right? They're the palatine tonsils. They're the palatine tonsils. But you also, at the base of your tongue, which is really hard to see unless these things are swollen, but at the base of your tongue, you have lingual tonsils. So this is at the root of your tongue, a pair of lingual tonsils. And then I want you all, I hate that I don't want to picture this. If you all were seeing me you are seeing me from the side, but if you were to like think about my throat, okay, so you're seeing me from the side, right? I want you to think about the back wall of my throat. Your throat's the pharynx, right? And then if you were to go up a little bit on that back wall, right behind the nose area, they're called the pharyngeal tonsils. People refer to these as adenoids.
There's a picture.
And there's, thank you, and then what page is that?
812.
812. And so I am going to want you all to look up and see where the location of these tonsils are. So let me tell you what tonsils are doing though. And look, let's see if it doesn't even make sense to you. They do have these little crypts, and y'all know that from the palatine tonsils that you can see so well, where they look like they have these little dips and valleys, you know? But these crypts aren't supposed to be like that. It's supposed to be that because that's how it traps potential pathogens that may be causing some trouble. And then look, you've got these nodules of lymphocytes. Did we say these white cells are going to be a good thing? So your tonsils are incredibly protective, especially when you're really young, because when you're really young, everything's foreign to you, right? You haven't, everything in your ecosystem is kind of foreign to you. So they're very, very active. So what you'll see is little kids' palatine tonsils. The other two are kind of hard to see, but the palate, just looking at somebody's mouth. But the palatine tonsils will be pretty big, and that's normal. Now they should not be red and inflamed, because that means that they're really fighting something, right? And certainly they shouldn't be abscessed. So I'm not talking about that, but they are supposed to be pretty big. But as we age, what normally should happen is that they involute. So by the time somebody is like 18, 19, their tonsils, their palatine tonsils should have shrunken so much that they're hardly even, they're hardly even noticeable. So how many of you had that happen, that your palatine tonsils did just what they were supposed to do? How many of you still have really huge palatine tonsils? Some of you do. Okay, you were the ones that were missed for tonsillectomies. Probably should have had tonsillectomies, right? Because what ENTs, I think, would mostly tell you is that these tonsils are one of our first lines of defense. They're doing an amazing job, and they usually are doing a perfect job. But every now and then, they're overwhelmed, and then you have tonsillitis. And if you have tonsillitis, that is literally means inflammation of the tonsils. Isn't that right? So repeated inflammation and swelling of the tonsils can kind of cause the tonsils to almost necrose. What does that mean? Yeah, so now instead of them protecting you, which they're still trying to do, they're actually maybe, because the crypts have gotten large, so large that they're now, instead of like catching and destroying, they're harboring microbes. You get what I'm saying? Yeah. So now they're actually more of a problem than they are of a help. And that's when the ENT has to make that decision. Is it worth taking them out? You guys, I will tell you the procedure, the surgical procedure, and I had an ENT tell me this, that I think I really respect their opinion. It's a much simpler procedure on a little kid, just because of the way the roots of the tonsils are, than someone that's older. Someone that's older, it is a much trickier, a little bit harder procedure, and it's kind of no fun. So I'm sure it's no fun for kids either. But I had mine out, but we remember pain. But I was little, kids don't understand what you said that. But I remember kind of having fun. Ice cream you can get? Are you kidding me? I would have done it again. So but anyway. But adults can hemorrhage more because the risk of hemorrhaging is a little bit higher. The soreness that they encounter, and it's probably a lot to do with just their inflammatory response, is heightened. As young adults, little kids have immature inflammatory responses, but as young adults, you have a good one, and you're strong. And so it's a combination of things that makes that a much tougher procedure on someone who's a young adult. Anyway, any questions for that? But are tonsils good or bad? They're good. They're good until they go bad. Then they're bad. Okay, that's time for a break because I'm obviously not making any sense. So 10 minutes. Let's take a quick 10 minutes. Okay. All right, so we said we have these tonsils. Let me tell you, y'all already know about the thymus, don't you? What does the thymus secrete, that group of hormones called? Thymosins. And we said thymosins are going to target T lymphocytes, and T lymphocytes are special kinds of white blood cells that are protected. So we said we get why in a child it's much bigger than it is in an adult, because as a child everything's new to the system, right? And then as our body's immune systems are maturing and we're learning all the things in our ecosystem, it actually starts to involute, and then by the time you're a senior person, it's actually being replaced by fat. So this is why the very young that are challenged and the very old are the most immunocompromised. Right? Young adults take a lot for granted. Okay, so anyway, we got the thymus. We know that. How about the spleen? It's part of your immune system. The spleen is. And does everybody kind of know where the spleen should be located? On the rats that y'all dissect, you're going to see where the spleens are, and on the rats, some things don't look like human comparative anatomy, but a lot of things do. And the spleen is one of the things in a little rat that actually is very similar to what the human spleen looks like. So it's going to be typically on this side below the stomach, but sort of below and sort of dorsal to the stomach. And this organ is going to be, in adults, going to be where blood reservoir is. Now, in fetuses, it can play a role in even blood production, like red bone marrow can, but we're going to just think about adults. So the blood reservoir It's also going to be where red blood cells are going to be kind of evaluated, and as they're getting older or they're getting misshapen, they're going to be removed from the system. So this is going to be where the red blood cells kind of go to die, if you will, and be removed from the cell system. So it's filtering, the spleen is filtering blood, storing blood, helping to remove old and worn out and irregular shaped red blood cells. So this is an area where it's going to be detecting potential foreign antigens, and this should say foreign antigens. So this is why there's some diseases that the spleen can actually become enlarged. What viral disease do you know? It's a viral disease that can cause the liver and the spleen to enlarge. What is it? It's mononucleosis. That is a disease that's caused from a virus called the Epstein-Barr virus. The EB, Epstein-Barr virus, causes mononucleosis. So this is one of the ones, but actually the spleen works in a lot as a daily kind of thing. This is why in when young adults have mononucleosis, their spleens can get kind of enlarged. And if the spleen is enlarged, is it more at risk for rupturing? Mm-hmm. Yeah, so young adults who have mononucleosis are told to limit physical activity for weeks and even several months after having active mononucleosis, because they are more at risk of their spleen rupturing. And because the spleen is a reservoir for blood, could they bleed out if it ruptures? Mm-hmm. Yeah, and it sometimes doesn't take too long. Now, can you live without your spleen? You can. So a splenectomy is a procedure that removes the spleen. It would be removed if there was trauma to the spleen and somebody was bleeding out, hemorrhaging in a quick hurry. And the only thing they could do is to go in there and just remove that organ to stop the bleeding. So in a lot of traumatic accidents, and sometimes they're automobile or falling or sometimes athletic, if the spleen is ruptured, it's an emergency situation and they'll have to remove it. That's all they have time to do. But if a person has had a splenectomy, they can live without their spleen, but they're forevermore immunocompromised. So they have to pay attention to making sure that they get all the immunizations, that they protect themselves, because they are going to be immunocompromised. Any questions? It's also why sickle cell disease. What did y'all tell me your red blood cells look like if you're in a sickle crisis? They're misshapen. And even when they're not in a crisis, they're still a little bit misshapen and a little bit harder. So the spleen on sicklers can often get enlarged. And usually, after even however many numbers of crises or even mild events, they're gonna electively remove the spleen. Why? Because it's painful, it's enlarged, and they're at risk of it rupturing. So this is why you will see sicklers that on their history, they've had splenectomies. And that would make good sense, wouldn't it? Maybe if that's the case, this happened. All right, so now I want to talk to you about some things. I want to talk to you about your immunity. And I got Kristen from Micro in here. I think Kristen's the only one in Micro. So sorry, Kristen, some of this will be repeat because we just kind of talked about a little of this in microbiology. But guys, we're born. This is referred to as innate. Innate means you're born with something, right? Innate or non-specific immunity, and immunity means protection. So you have two types of protection I'm about to talk to you about. I'm going to talk to you about what you're born with, the innate, non-specific immunity protection. And then afterwards, I'm going to talk to you about specific or adaptive immunity. And I need for you all to be kind of conversing about this because you'll have quite a few things on this test about these two different types. So when we think things that you're born with, what are you born with that protects you? And I know it's probably right up here behind me. Yeah. Okay, so what are you born with? You're born with skin, right? That's our largest organ, and our external barrier is our skin. It's waterproofing us. There are microbes that live on it that protect us, that provide like this acid invisible shield that we have. It's weird to think about that we walk around with this little invisible force field, isn't it? That because of the microbes that live on us, but we do. So our skin is protected. How about tears and saliva? Is our food sterile? Our food or drink sterile? No, that's why there's that 5-second rule. So you can pick things up and it's fine. But as long as there's not too much stuff on them, we know that we're not going to, I mean, we don't have food poisoning every day, do we? But guess what? Almost every day we swallow the microbes that could cause food poisoning. We have amazing protection, don't we? Because in our saliva, we have enzymes that break down microbes. Our stomach acids take the numbers way down. And food and liquid does not have to be sterile. So saliva, gastric juices, how about just the acts of urinating and voiding? Isn't that protected? They really are, because when you're defecating, you're actually moving things that are undigestible through your system and keeping them off the healthy tissues or the inner lining of your colons. So just the act of defecating is kind of protective and getting rid of things you can't digest. The act of urinating is cleaning out the opening of the urethra, because the opening of the urethra is actually colonized with microbes, and that's normal. But you wouldn't want those microbes being able to come up through the urethra, would you? So you void several times a day. And so just some of the things we do. Blinking keeps tears coming in our eyes, and our tears actually antimicrobial. Do they have antimicrobial substances? They do. They do. How about sweat glands? Same thing. So these are all things you're kind of innately born with. How about give me something else that's not up here? Think of something else.
Is that like the thymus? Or no, that's
Well, the thymus is going to be about those T lymphocytes. We're going to talk about in a bit. But how about just the act that you can have a fever, that your temperature can change? That's a pretty good trick, isn't it? That's a pretty good trick, that you can raise your temperature. Because when you raise your temperature, is it going to discourage some microbial growth? Is it going to increase your metabolism so that your healing will be faster? Is it going to bring more circulation faster to the sites? It is. So having a fever is really protective. Do you just have a fever in a particular disease, or do you have the ability to do that quite frequently if you need it? Your body needs to. Do you know what controls your thermostat? What part of your body? Because you would want to know. It's your hypothalamus. Controls your thermostat. So your hypothalamus is getting signals. Didn't we say the hypothalamus is kind of the seat of your universe, right? So it's actually controlling that, and it gets signals that maybe tissues are damaged or maybe something's invaded, and it raises your set points, your temperature set points, and that's very protected. People should leave fevers alone up to a certain point. Now, not many people do though. Most people, if there's any sign of a fever, what do they want? They want an antipyretic. They want to take something that's going to bring your temperature down. But actually, in a perfect world, we would really watch them. And as long as we see a new set point that we're vacillating around, we will leave it alone, because it's really protected. How about inflammation? The inflammation gets a bad name just like fever does. What are the 4 cardinal signs of inflammation? Swelling, redness, heat, and pain. So those are the 4 cardinal signs of inflammation you should know. And are those things bad? No. Those things are all good. The swelling is because more blood's coming to the site. And with more blood, it's going to be white cells to protect, right? Nutrients to help with tissue repair. And is that true? And the heat is going to discourage any microbes. And it's the blood that's bringing the heat, right? So all those things go good. Well, how could pain be good? So the pain is good, so you know that you have an injury there, so stay off of it until it heals, right? Or take care of it, whatever you got to take care of it. So those things are all good up to a point. Can we have too much? Yes. Yes, we can have too much. And if inflammation is too much, can it then start to damage? Yes. Yeah. But the problem comes in who's assessing how much is too much. And most of us just don't want any inflammation at all, because we don't want to be inconvenienced, and we want a pill to fix everything immediately, right? So that's where kind of the problem comes from. It's normal to have inflammation, and it's protective to have inflammation up to a point. Okay, so good. Now these are all just things. I want to tell you about some other things though too that are non-specific. And I know I'm doing it from one slide, but you guys have, well, actually, Actually, I'll go ahead and make sure. You have, we just talked about, you have circulating neutrophils as adults. They're the predominant white count. Don't you know? You're going to have to know that from the test. So you have neutrophils that fight against bacteria. Is it just one type of bacteria or just any? Any. So that's kind of non-specific, isn't it? We have those circulating. How about eosinophils that have histamines? They also are going to be protective. And we said we're going to see these increasing if there's allergens or worms, parasites, didn't we say? We have basophils that have histamines and heparin, an anticoagulant, so that, these are going to protect us too in our tissues. Lymphocytes, we said, are going to fight against viruses, just one virus or any kinds of viruses, and even cancer cells. So we said also it's kind of normal that cells are losing their life cycle controls at any given time. But that's okay, because we have these lymphocytes circulating around that are kind of non-specific. The ones that we really get a lot of attention are the lymphocytes, because y'all are about to hear about different kinds of lymphocytes. We've already hinted that there are different kinds because we talked about T lymphocytes. And they're called T lymphocytes because they're dependent on what organs? The thymus. So you've already kind of heard that maybe there's different kinds of lymphocytes. Here's one. It's called natural killer lymphocytes. These guys are no joke. These guys are the lymphocytes that are circulating around just looking for something that's not self-antigens, something that's foreign. And they don't care what it is. Guess what they're going to do? They're going to attack it. They are.
Is that what, for autoimmune diseases, is that what attacks your?
In some autoimmune diseases, in some, we're going to talk about that. All right. So monocytes, didn't we say these are going to be our big eaters? They're going to end up aggregating to make macrophages. They're going to clean up debris. They're actually going to have more functions than that. They end up being what are called antigen-presenting cells. You don't really need to know that, I don't think. But anyway, they're going to show our system when something foreign comes in. These monocytes will have eaten it. And like if this is something foreign that's come in, these monocytes are going to eat it. I'm the monocyte. I'm going to take off its little foreign antigen and put it out here on my membrane so my other white cells can see this is foreign. Attack this. Because every single foreign pathogen has unique outfits. And these monocytes, as they eat them, they present that out so that the rest of the immune system response can respond specifically to that. We'll talk about that later. But anyway, monocytes act as macrophages. We said inflammation, we know it's good. Now white cells, they're going to be different things, different chemicals that are going to be put off by our tissues as they're being damaged. And what you can really think about these, and I don't think I asked other than histamine, I know I asked you about histamine, but leukotrienes and bradykinin, we have these alarm molecules. They're like sirens, right? And so what these molecules act like is actually alerting these cells to actually come to the site and get to this site so that healing can begin, or the process of the healing can actually begin. So really, essentially what these are these molecules that are going to be released to actually call things to the site. So I think this is a pretty good little illustration where you see a splinter coming in, and splinters are rarely sterile, are they? They're not. Well, your skin's not sterile. So even if you did somewhere in the world find a sterile splinter, your skin's not sterile. So as it entered your skin, it just introduced stuff into the system. And so now you've got all these inflammatory chemicals are being released by the damaged tissues, and they're going to be coming to the site and calling other cells to the site so healing can actually happen for tissue repair. Now, I want you to know about some antimicrobial proteins. Our cells are essentially what kind of factories? Protein factories. So these are very specific kind of proteins, and the first one I want you to know about are interferons. Interferons are going to be these proteins that our cells produce when they become invaded by viruses. Now, the crazy thing is you're thinking, okay, well, maybe the cell produces these interferons to protect themselves from the viruses that just invaded them. But no, that's not really the case. And so listen so that you don't see a textbook and not know exactly what's happening here. I want you to think about your nasal mucosa. Oh, that probably didn't sound too good. But anyway, but think about air moving in, right? And so viruses enter the nasal mucosa, and some whole viruses will attach to some of those nasal cells and can cause a cold. Isn't that right? Is that right? Is everybody picturing? Now, the reason that you don't get sick and die from colds is one of the reasons is that we do know that the cells that become invaded by viruses, they're very altruistic in like the best sense of the word. They're going to produce these interferons that protect neighboring cells. It's almost like houses in your neighborhood. Robbers come and rob one house. Now that house is going to be robbed, and maybe there's going to be murder and mayhem in that house. But before that, the people in that house die, they're putting out alarms, and all the houses in the neighborhood are shutting their what? Doors and windows. So that's how interferon acts. A cell that's invaded releases this interferon, and neighboring cells shut down their doors and windows so the viruses can't. It limits the attack.
So it's like an alarm.
It is. It's like an alarm. I gave two analogies. Do they make any sense? Okay, so this is how interferon. Like, I remember this in medicine when we were first realizing what interferon did. And the thing was, it was thought, because it also has anti-cancer types of effects. It was thought that interferon, because we were able to, with genetic engineering, start to make this in the lab rather inexpensively. And it was thought this was going to be the magic bullet that was going to treat all serious viral infections and all cancer. What an amazing thing, right? This was going to be this magic bullet. Now, has it turned out that way? No, not exactly, because everybody's different. We know cancer is not just one thing, is it? It's many, many different things. And even the same cancer in two different people can act very differently. Yeah, so we know this is way more complicated than social media makes it sound, right? So we understand that. But is interferon being used? We actually can make it now, and it's being used for like hepatitis C, which infects the liver, those types of serious viral infections, but also as anti-cancer effects, and it can actually slow it down. So it's been useful, but it hasn't been the magic bullet we wanted. So interferon is one. Here's some other proteins. They're called complement proteins. I need you to know this. I need you to know that these complement proteins, and by the way, these complement proteins have been numbered. Complement protein 1, complement protein 2, 4. They've been numbered by when they were found. So the third one was found when? Third. It was the third complement protein found. Now, you don't have to know when it was found. I was just telling you why they're named the way they are. Now, here's the thing about these complement proteins. Where do we inherit our protein information from? So if we got these complement proteins circulating around, just waiting for something to happen for us to be invaded by pathogens, then they're in place. And this is all part of stuff we're just born with, because we got this from our parents, our DNA. But if we got them and they're present, and we get invaded by something that's potentially pathogenic, do y'all remember me talking about the blood clotting proteins will start a cascade kind of effect if they become activated? That was the intrinsic and extrinsic right, pathways. All right. Well, these kind of do it the same way, where these proteins, these complement proteins, if they get a signal, they're going, and I'm going to tell you what sends that signal in a minute, but when they get a signal, they're going to start interacting with each other, and there are going to be 3 potential outcomes of the complement proteins. I talked about inflammation, didn't I? Did I say that's a good thing or a bad thing? That's a good thing. So we know we have inflammation because of what? Complement proteins. So we have this inflammation, and that's a good thing up to a point. But the second thing I want you to know is called opsonization can happen. If these complement proteins become activated, they actually, if they see something foreign, these complement proteins will start packing on it so that the monocytes can easily phagocytize it. So opsonization is literally a coating of the foreign antigen or bad guy, pathogen, so that the white cells can do their work by eating them. Okay, so it's the same as if I told y'all to all stick out your tongue and I put a little dry aspirin on the end of your tongue and said swallow it. Would that be easy to do? But what if it was a capsule, like a gel capsule? Would that be easier to swallow? That's exactly what opsonization is. These complement proteins literally do that. The third thing is called cytolysis. If I broke this word down, what does cyte mean? And what does lysis mean? To split a cell. So what these complement proteins do in a foreign cell is penetrate the wall of the foreign cell, and as soon as they have all lined up in place, what is now the wall looking like? It's got a what in it? Right? It's got a hole in it. And so now this has lysed that cell, hasn't it? So 3 outcomes of complement protein activation: inflammation, cytolysis, and opsonization. Are we good with that? What would happen if you were born without information for some of your complement proteins? You could be compromised, right? You could be immunocompromised. Some people actually are born with that. All right, so we said fever is a good thing, isn't it? And getting back to fever, let me tell you what the chemicals that are released in the body by damaged cells. These chemicals that are going to be released by the body are called pyrogens. Pyrogens. What is a pyromaniac likes what? Fire. Y'all know that, right? So pyrogens are going to be these chemicals that signal the hypothalamus to do what? To raise the body temperature. So what is like ibuprofen and acetaminophen and even aspirin? What do they do? They're anti-pyretic, aren't they? They're going to bring that back down. Now, I want to let you know about some terms that you might hear about. If you have normal body temperature in Celsius degrees, 37, you're vacillating around it, but then you become infected, right? Something that's going to end up causing pyrogens to be released. The hypothalamus is going to reset your thermostat. This new area that you're vacillating around is called the stadium. And then after, hopefully, your body kind of figures out and controls the infection, you're going to return back to normal. When you're returning back to normal, people say the fever broke, right? You hear about that. But that's actually the term for that is defervescence. Defervescence. So I want you to know stadium is referring to that new thermostat setting. And defervescence is meaning you're returning back to your normal temperature. Good? Are we good? All right. Now, I want to talk to you about everything you just mentioned are things that you should be born with. And what kind of immunity did I call that? Innate or non-specific. Exactly. Innate or non-specific. Now that's all beautiful and that's really good. But if that's all you had, you wouldn't make it out of infancy. We want all of it. We want that. We want to have all of that because we know that if we don't have that, we're going to have a little bit of more of a time struggling to fight this. But if you didn't have this next part, if people didn't have this, some people don't have very good at this next part. But if you don't have it, you don't make it out of infancy. So I need you to be paying attention to what I'm about to tell you about specific immunity. There's another term for it. We called it what? Adaptive, meaning that you can adapt to something in your environment. Is that right? Now, what I'm going to want you to know is that there's 2 types. There's 2 types. Now, these 2 types, they're going on at the same time, but I can't talk about them at the same time because I'm not that talented. So, but I'm going to tell you about them one at a time, but you're going to understand that they're happening at the same time. And what is happening is this, a specific immunity. When you get exposed to a foreign, it's not you, it's not your cells, it's not yourself. When you get exposed to a foreign antigen, do y'all remember what antigen, what I said antigens really are? Oh, Lord have mercy. A foreign, and I want that to look like a diamond, but anyway. I mean a triangle. Well, okay, so what sorry. Okay, so, well, look, something just foreign came in. It just maybe came in through your nose, maybe you swallowed it. I don't know what you did. Put it in your eye. I don't know. Maybe it came in through another place. But anyway, it just entered the body and it's foreign. And remember, antigens are proteins, right? We have our self-antigens that we should always love, our self-antigens. But we said, y'all said you remember there's a protein that can be made that fights antigens. And what was that protein called? That attacks, annihilates, destroys antibodies, right? So you're talking about proteins, which are just molecules. They're not alive, are they? So there's these antigens, but then our body has the ability to make a complementary protein called an antibody that will be specific against that particular antigen. Should that antibody actually cross-react with anything else? No, because it's supposed to be what? Yeah, so then it usually is. So let me tell you what's going on with humoral immunity. In humoral immunity, it takes a special kind of cell for humoral immunity. They're called B lymphocytes. B. That B stands for bursa, which is a special types of tissue that these are kind of, well, anyway, just know they're called B lymphocytes, it's fine. Now, if the body signals that a foreign antigen has entered the body, an inactive B lymphocyte, and I didn't put much space into there, that's a B lymphocyte, an inactive B lymphocyte will be given a signal to respond to this foreign antigen. And this inactive B will undergo mitosis. What does mitosis mean? Cell division, is that right? Cell division, and as it undergoes mitosis, a lot of these cells will be made. They're called plasma cells. These plasma cells are going to produce a protein called antibodies, and those antibodies are going to have the exact specific shape to take out these what? These antigens. Antibodies are proteins. Antigens are proteins that the antibodies are going to be directed against. And they're going to seek them out like little missile-seeking molecules. And when they get to those antigens, they're going to take them out. Are you with me on that?
What does the plasma cells have to do with it again?
The plasma cells are what are producing these antibodies, these specific antibodies. These B lymphocytes take in these antigens, study them, become active, and then undergo mitosis. And they start dividing into plasma cells that have one function. The only function this plasma cell has is to make an antibody that's specific against that antigen. Are these plasma cells going to last long in the system? No. Just long enough to make a ton of this, and as this increases in amount, this is actually referred to as, you can do a test to see how much of it's there. It's called a what? A titer of antibodies. And as the amount of these antibodies are being released, what are they attacking? Antigens. And as they're attacking the antigens, are they removing them from the system? And so signs and symptoms of the disease should be going what? So as the titer of the antibodies go up, which is the quantity, the amount, are going up, signs and symptoms are going down. Are you with me on this? Everybody good with this? Any questions? You're going to have to know this pretty well for that test to be able to answer questions on that test. And if you're going to be looking, because I know a lot of you are so entrenched in seeing verbatim something in the textbook, because that's the way K-12 has done that to you, you're going to be out of luck, because that's not the way it's going to be written. So I hope everybody's listening and staying, because even though it's going to be open book and open notes, you're still going to have to know it pretty well. You're not going to find something verbatim in the book.
So the titers are what you draw to, what are the titers for again?
A titer just simply means the amount. of these specific antibodies, the amount.
Okay.
You just mentioned something that you can draw someone's titers, and you can draw blood and look to see how much, the amount of a particular antibody somebody has circulating around. And they are called titers for that particular antibody. You can. If I wanted to see if I've ever had chickenpox before, varicella zoster is the virus that causes that. If I'd ever been exposed to that virus, if I ever had, I would have made an antibody response to it that are specific to varicella zoster. So I could actually draw blood to see if I had titers to varicella zoster. I could do that, right? And how much I have of that. I might not have any. If I don't see any titers are non-existent, it means I've never been exposed to what? To chickenpox virus, varicella zoster, right? If I've had the hepatitis B vaccine series of 3, right? If I had that hepatitis B vaccine, which is against the hepatitis B virus, if I've had that vaccine after the 6-month period, wait a little bit, I could look to see if I have titers against it, to see if I actually built a response. And if I have titers against that, if I have antibodies, does it mean I'll be protected if I ever get exposed to it? It does. It means I will be protected. So hold on with me for a little bit, and I hope as I go along, it might answer some questions that you might end up having. The beautiful thing about this initial disease thing that you get, because you've never been exposed to this foreign antigen before, not ever. And the beautiful thing is, you know, after you get exposed the first time and you start to build an antibody response, the beautiful thing is there will be a very few cells that are made that are called memory B cells. Memory B cells. These memory B cells are going to always be present the rest of your life. And so you will never get the signs and symptoms you had the first time, because these are going to be that's all the function they have in your life, is to just look for that particular antigen. If they see it, they're going to immediately be able to produce high titers of antibodies, because they don't have to study it, they don't have to undergo anything. They are already going to be present, and you're going to produce antibodies so quickly that those antibodies are going to smother those foreign antigens, and they're not going to be able to attack anything. I give the example because I think it's a good example to give of my kids I had 3 kids under 3. Well, one had just turned 3, but the baby was 6 months old. So 3 babies, and they all got chickenpox at the same time. And that they had, I mean, when I say hundreds, I'm not exaggerating that, hundreds of little pox, you know. And, you know, and chickenpox is a respiratory-borne illness. So, you know, they're snotty and fevery and pocky and, you know, and they're babies, so it's not everywhere. There's, you know, a lot of bodily fluids. So whatever. Do you think I wasn't wiping varicella zoster in my nose and swallowing it and putting it in my eyes? And you don't think it was getting on me? It was all over me. It was all over me. It was in my lungs. I was breathing it in. Did I get sick? Why not? Because I had it years ago. I don't even remember having it, but I had it years ago, and I had memory B cells that started pumping out massive quantities of antibodies within minutes. So that it didn't matter how much I was inhaling or swallowing or whatever, were my antibodies taking them out. That's what kind of immunity? Now let me say this about you guys and us. That's just with something, you know, not that but that's something like chickenpox. But that's with everything that's foreign in your environment. You have made this kind of, and this is only half of it, I'm talking about the other half. You have made that kind of response. So as long as you're talking about something that has a stable antigenic structure, you will never get it again. Now, do all things have all foreign potential pathogens have stable antigenic structures? No. So for example, the influenza virus that goes around each year, this year it might look like this. And so you may get this virus and you have to produce an antibody that looks exactly, complementary to it. They can take it out. You get sick for a week to 10 days, and then you're going to get better. But then next year, you know what it does? Next year, it changes its outfit. And then it changed the antigenic structure. And so next year, those antibodies don't work against this. And so what did you got to do? You got to get sick again and make a new antibody. Do you get what I'm saying? Do you get what I mean? So there's some things that shift, antigenic components, they're called. But most things, thank goodness, are stable in our system. So once we have them, once we learn them, we never had them again. Isn't that kind of beautiful? Right? So we don't have them again.
Do you have to actually get sick to produce those
No,
antibodies?
no.
Okay.
Most things, thank you. No.
Like you can
So
have
little
exposure.
kids, because little kids are, I mean, imagine a baby going through the birth canal. Babies going through the birth canal, they're getting exposed to all kinds of foreign things, right? And most things don't make you sick. Most things don't make you sick, but do you need to learn them? Yes, you do. Your immune system is being challenged and you're learning them. So for example, an autoimmune disease, type 1 diabetes. Type 1, insulin-dependent diabetes mellitus used to be called juvenile. Less than 10%, about 10% of all diabetes mellitus. That's an autoimmune disease. And it usually does happen young in life because it's usually from a virus that you got introduced to that didn't even make you sick, but it caused you to make what? And those antibodies were so close in their structure to your beta islet cell antigens, the beta islet cells in your pancreas, that they cross-reacted with them. Do you hear what I'm saying? And they cross-reacted with them, and then it destroyed the beta islet cells. But what you had never even made you. Most things don't make us very sick because we're starting with this. Now, there's some things that we can get pretty sick with, and who's had chicken pox that they actually remember having chicken pox? Some of y'all remember having it?
I was 18.
So, oh my gosh. Okay, so you were miserable. And so those, whoever raised their hand that remembered it, you didn't feel good, did you? You were really sick because that's called fever systemic. Chicken pox is a respiratory-borne disease. Do we ever get rid of the virus? No, because it can lie in the neurons, in the dorsal ganglia for years. Now, most people will never have a reoccurrence of shingles after that, but it can cause shingles as a secondary thing, and about a third of those people who have chickenpox can develop shingles later. And then on rare, shingles is not respiratory-borne though. My son the other day is down in Roanoke at Brilliant, and they had put a patient in respiratory isolation because the patient had shingles. So he's texting me, and he said, shingles is not respiratory borne, is it? You told us that it wasn't. I said, no, it absolutely isn't. They need their IV before. You can't catch shingles from somebody. You can't, if you've never had chicken pox and I have shingles, I can't give you chicken pox because the shingles, now the virus is on me, but if you cover up those lesions, it would take contact. That person should have been put on contact, and he knew that, family knew that. They should have been put on contact isolation, but not respiratory. Do you get what I mean? Because a person who has shingles is not breathing it out. A person who has chicken pox, that is respiratory. Am I making sense? Mm-hmm. All right, so anyway, but those kind of things need to be known about all of it. So now I'm talking about micro, and I need to change back to A&P, but are we good? So anyway, this is half of what's going on in your specific immunity. Now let me tell you what's happening on the second half, and that's cell-mediated, because this is pretty cool too. These things have just entered the system, and they're also going to be alerting and cell-mediated a special kind of cell called a T8 cell. This cell is also on lab reports, called a CD8 cell. And on some lab reports, it's not my fault it has so many names, so don't blame me for this. On some lab reports, it's also called a cytotoxic T. And usually that'll be abbreviated. But that's all one cell. That's all the one same cell. It's just different names, different labs will call it, okay? These cells, do you know why they're called cytotoxic? don't confuse them. And they're called T's because they're T lymphocytes. So what are they going to be dependent on to actually mature? Thymosins from the thymus gland, right? Yeah. And so now these are not the same as the natural killer cells. They're not lymphocytes. These are very specialized kind of cell. You know what I call these cells? These cells become active. Now it takes them the first time you get exposed, and the first time they see this little guy, look, they're looking at him. The first time they see him, it's going to take them a little while to figure out the structure, and then they're going to become active. And I'm going to tell you something. You know what I think about, and I really think it's a great analogy, and I don't know who could forget it after my analogy. If you think about this protection, this specific immunity protection, speaking like our military, this is our military that's protecting our country, or anybody's military is protecting our country. These B lymphocytes that become active and become plasma cells, they're like the Air Force. And the Air Force is going to drop what? Bombs on the enemy, right? Is that right? Is that what the Air Force is doing? Dropping bombs on the enemy, hopefully pulling them up, right? But the cytotoxic T's are the Marines. They don't get bombs. And those Marines, guess what they learn to do? They're called riflemen, but guess what they really learn to do? They really learn to kill people with their hands. It's really sad, but that's what they do as long as they're not in the office somewhere. They learn to do that because it's cell to cell. And that's what these guys are. Okay, so they're going to, wherever there's a foreign thing that has this little antigen on it, they're not going to send a bomb to do their work. They're not going to do that. They're going straight to it. But the whole idea is, are you taking these things out? These cytotoxic T cells are taking them out. The bombs are taking them out, and they're so incredibly effective, right? And you know what's the beautiful thing about these? After the initial, guess what you get? Memory T cells too. So the next time that same antigenic structure that's foreign enters into your system, does it take these memories long before they have to discover what it is, or are they just waiting for them? They just are waiting for them, and so you don't get things twice. You don't. I know some of you said, well, my mama told me I had chickenpox twice. No, you didn't. You probably had chiggers. You probably had chiggers once and you had chickenpox once, but you didn't have chickenpox twice. It's never been documented that that's been shown. Now, immunizations. For immunizations, when you get an immunization, are they giving you the actual pathogen? They're not giving you the pathogen. There's different ways that they do this. There are different types of immunizations. But what they're doing is just presenting the outfit. Did the outfits cause you to get sick, or is it a pathogen? In immunizations, they're presenting the antigens, those protein antigens, so that you make the same responses. Now, it can take a while, just like if you got exposed to them in nature. You know, it takes about 14 days or so to build immunity to get this kind of protection. But if you get an immunization, is there any way to get the disease from the immunization? No. How many of you have heard that you can get the flu from the flu shot? How many of you have heard that?
I've heard the symptoms of it, but not like.
You can, let me just tell you, if you ever take an infectious disease class, you'll learn about different diseases. And when you're learning about the different diseases, my micro people are usually saying, well, God, every time they say a new disease, it says flu-like symptoms. So flu-like symptoms. What are flu-like symptoms? Malaise.
Body aches.
Yeah, so body aches, so you're tired, you might have some body aches, you know, you just don't feel good. So, you know, that's flu-like symptoms. You cannot get it. It's impossible to get the flu from the flu shot. You cannot get it from the flu shot. It cannot. Talk about divine conception or whatever. That would be, because you can't get it. And you can get it from the person behind you in line who had the flu, because the flu shot's going to take about 14 days, 14 to 21 days before you'll be protected. So somebody behind you in line was starting with incubating the flu. They're not as sick yet, but they're like, they just on you. So, but I had flu shot. But anyway, if they did that to you, could they actually give that to you and then in a few days you get sick, but you think it's because you got the shot? Yeah, you did. Or you got it in line to the grocery store, or you got it when you pumped your gas from the gas tank, or you got it when you turned the knob on the door in the bathroom. But you didn't get it from the flu shot. Now, can preservatives that are found in immunizations cause some of those symptoms? Are they usually kind of mild, those symptoms, if you're going to have a response? How many of you had adverse responses to immunizations? How many of you have ever had adverse responses? It's kind of rare that people have adverse reactions. Usually it's local, and it can be like a local thing, and that's usually just because of how it was given. Or, and sometimes it can be kind of systemic, flu-like. You know, you can feel a little bit, but you don't know if that was from that shot, or you don't know if maybe you had something. But it wouldn't have been from the shot. You wouldn't get flu from the shot. It is impossible. Okay. Any questions about that? Now, so you're thinking to yourself, okay, so who's the first to see this little foreign thing? And let me tell you who the first is. And this is just so important. I'm going to write it in red. There's another kind of cell. So far, we've talked about this cell and this cell. This cell being part of humoral. Humors mean the fluids, by the way, the fluids of the body. And antibodies are released into the fluids of the body, extracellular secretions, right? This is cell mediated because we said these guys aren't releasing anything, are they? What are they doing? They're cell to cell fighting these guys. So we've got this great thing going on. But when these foreign guys first enter the body, what alerts these inactive Bs and inactive Ts to become active? There's another kind of cell, and these cells, and again, not my fault, they have so many names, but it's the same cell. It's the same cell. It's just different ways. You'll see it on the lab reports, depending on the lab. CD4, T4, or sometimes referred to, and I like that when it's referred to as this way, helper T. And again, these are T lymphocytes, aren't they? These are lymphocytes. These are helper T lymphocytes. All the same kind of cell, just different names depending on the lab. These helper T lymphocytes are circulating around, and they are your first alert that something foreign has entered the body. These are the cells that are actually going to produce these leukotrienes, these chemical signaling molecules, to tell this cell to become active and to tell this inactive one to become active. These are also going to be these chemicals that are being produced by the helper T lymphocytes are also going to activate inflammation. So the complement proteins, even though that's non-specific, it's still doing that. So do you see that your entire specific immunity, which includes both humoral and cell-mediated, is hinging on one type of lymphocyte, the helper T lymphocytes. It's hinging on them. Because without them alerting and letting the bees tell them to become active, we've got an invader here. You know what the helper T lymphocytes, the CD4 lymphocytes are like? They're like the Pentagon, right? You can think of it as that way. They're like the Pentagon. So everything hinges on it. Do y'all, when we think about that and we say without specific immunity, we don't really get to last long, do we? We really kind of don't, because we gotta have this going. Do y'all know what virus attacks the helper T lymphocytes? HIV. The human immunodeficiency virus is so devastating to your immune system because it attacks these cells. Does anyone ever actually die from HIV, that virus? Huh? No. That virus itself is not going to cause you to die. But that virus, because it's taking down the numbers of your helper T lymphocytes, it causes you to not have a strong immune system. So then something else like a fungal pneumonia, which nobody gets a fungal pneumonia unless they're immunocompromised, or something like herpes that should just be causing genital or oral lesions now goes to your brain, right? And that's the end of the line, usually. So you end up getting, because your immune system goes down so much, you end up getting opportunistic infections that take you out. Are you with me? The virus, that virus itself doesn't kill you, but it's really weakening the immune response. You should have more than 600 of these cells circulating per microliter. But what they'll do with people who have HIV positive status is routinely check those counts. So if you see CD4 counts or helper T counts on somebody's lab work, is that they're watching the numbers of those. And once they go below like 200 cells per microliter, you're also, it's correlating beautifully, or not beautifully, it's actually tragically, with opportunistic infections happening. And once you get below 200, they say that you're then in full-blown AIDS. What does AIDS stand for? Acquired immune deficiency syndrome. Acquired means that you caught it. Don't anybody in this class tell me that AIDS stands for auto. It doesn't. It's acquired. It's an infectious disease, isn't it? Acquired immune deficiency syndrome. Know what AIDS stands for. If you have HIV positive status, is it a given that you will develop into AIDS? No. What's going to help you to not? What you said? What? Medication. Taking your treatments. Now we have people who are living normal lifespans with HIV positive status that they've had for decades. They're living normal lifespans. That doesn't mean everybody's going to get to do that, but early detection, proper treatment, and people are managing this and not developing, going into that, because it is the immune system devastation that ends up causing death for people who have HIV positive. Any questions from me? So these helper T cells, by sending out these chemical signaling molecules, activates humoral immunity. They activate the cell-mediated immunity. They activate the complement proteins. They're really, no wonder they're called helpers, right? The helper T lymphocytes. So we have helper T lymphocytes, we have cytotoxic T lymphocytes, we have memory T lymphocytes, and we have one other kind. And these other kind are called suppressor T lymphocytes. And guess what they do? They suppress the entire response. And let me tell you why you have those. Just like, and now I'll skip from military, but in the military you have something like this, maybe police, because people have heard about, without police, what would happen to society? What would happen to society?
Crazy.
You guys, we don't even want to know, because it would not be good. It would be, I'm not saying that the police are perfect. They're not. The militaries are not perfect, and people aren't perfect. But without it, it'd be total chaos. Have y'all seen what happens when we just had floods in areas? What happens? Societal norms break completely down, don't they? And people do crazy things that I don't think they necessarily would do with some controls in place, right? So we have to have the police. But in every single police department, there's always one person that's the internal affairs person, designated person. So your suppressor T cells are the cells that are keeping those cells in check, but they're not stepping out of line. When they do step out of line, what can they cause? When your own white cells are attacking yourself, autoimmune. That's exactly right. And I do want you all to know about suppressor T lymphocytes, because now that we've known about them for a long time, but a lot of research is going into suppressor T lymphocytes now and helping to figure out how people who have autoimmune diseases, it might be because their suppressor T cells are not doing the job that they should have been doing, and we can maybe use and harness them to help that person with autoimmune diseases. Does that make sense? There's another thing that they're called, it's just another name, and I never call them that. I always call them suppressor Ts, but they're, it's another word that's sort of like suppressor. But I don't know, I call them suppressors. So if y'all heard that other word, you would know we're talking about this class. Yes?
With AIDS, could you get like the flu shot or something like that, or would that compromise your system too much?
No, if you're immunocompromised, you should make sure you're getting people who are immunocompromised for whatever reasons. And by the way, I want y'all to write down. There's two types of being immunocompromised. There's two ways that we can be immunocompromised. We can have primary immunocompromised states or secondary. Primary means that you're born with something that's causing you to be immunocompromised. I'm sorry, but I just want what it is, but I'm not that. So primary means you're born with something. So like you don't have maybe a thymus. If you're not born with a thymus, what can't you make? Thymosin. So what is not, look at all my good T lymphocytes, guys. You see that? And so you're not making, are you kidding? They're not maturing. If you're born without a thymus, you're put in a bubble. And back in the day, you just wouldn't have gotten out of infancy. You would have died. Have y'all ever heard of the bubble babies? You haven't? You have. Well, so they're the ones that get put in, if they don't have a thymus functioning babies, they get put in, they can never have human touch. Their food and liquids have to be sterilized. They can't survive in the world. So before actually a bunch of them are making it out now because of stem cell replacements. It's still kind of new and whatnot, but some of them are making it out successfully. Now some, because the alternative is they live and die in a bubble. But some of them have made it out and living successfully. Some of them, when they got the stem cell replacements, ended up developing leukemias at higher rates than normal population. So was it something that probably generated that? Yeah, probably. But the alternative of staying in the bubble, whatever. So hopefully it's getting better. But that is kind of a rare thing. But you could be primary immunocompromised because maybe you're missing one of those complement proteins. Now that would be a much lesser kind of thing, wouldn't it? Or you could be one of your antibody classes. You're not making those. I haven't talked to you about those yet, but maybe you're not making one of those gamma globulins. And so that's going to be something. And it's going to be a lesser type of primary immunocompromised state, but it's something you're born with. It's usually genetic. Now, secondary immunosuppressions and immunocompromise are acquired, aren't they? Now, obviously, infectious diseases can cause you to be immunocompromised. And the one that gets all the notoriety is HIV, right? So the human immunodeficiency virus that ends up attacking these very particular types of cells that wipe out your specific immunity. Before the treatments, most people with this did develop into AIDS and did die young. Now, we have come a long way. We haven't come as far as we need to, so hopefully this is one of those viruses it's really difficult for us to get a vaccine on because it's changing its outfit. And actually what we're seeing is that even one person might have several different types of HIV virus. They've been exposed to several. Is HIV virus an easy virus to transmit? Yeah. Is HIV an easy virus to transmit? But that's not so easy.
But it's like unprotected though. I mean,
no. Okay, so you guys, about certain infections are very easy. Like measles is respiratory. Chickenpox is respiratory. This is why measles is really, people should be being concerned right now. Not just because they may get measles or their child may get measles and be one of the people that develop into encephalitis that die or have lifelong neurological consequences. Because if measles are in the population, you have a young person who is 18 and pregnant, it's devastating to a fetus that she's carrying. So, some things are respiratory that are really easy. Hepatitis B virus that attacks the liver lives on surfaces a long time. So you would have to have either body fluid contact or it can live on surfaces that haven't been cleaned for a long time. HIV does not live on inanimate surfaces, just seconds. It does not live on them. And you have to have a certain inoculating, an infectious dose it's called, a certain inoculum or an infectious dose of it. It's not even enough of it in saliva to be transmitted. It's not. You have to have a direct fluid contact. Now, of course, it's not hard to have sex, which is a good thing. But you would have to have done that. You would have had to have gotten blood products, shared needles, had sex with somebody who had it, right? And even having sex, there's certain types of sex, because oral sex is sex, isn't it? I think a lot of our young people don't get that memo. They should get that memo immediately, like young, before they even start the curiosity thing. Because a lot of them say, no, I'm a virgin, I only had oral sex. Okay, what? What? No, that's adult behavior, and all with adult behavior comes all the risks and responsibilities, right? So there's no stigma associated with sexually transmitted diseases. Anybody who ever says a sexually transmitted disease name that has any kind of connotation that sounds like a stigma is uninformed and ignorant. There's no stigma associated with that. We're all here because of what? Sex. As a certain part of our development, what should we all be doing? Having sex, right? Because it's natural. It is a natural thing. It would be the same way as if somebody said, she's got that cold from that kindergarten that she was it's just crazy. We live in a microbial world. We live in a world where, as humans, we have certain developmental things that are normal and natural. The reason that we have so many sexually transmitted diseases out there is because there's ignorance and stigma associated with it. If all young people, before they started that curiosity thing that keeps them, doing things that, before they start that, if they just understood that responsibility, so that when they started with maybe a sign or symptom of something, they thought, oh, the same way that if you have somebody with a sore throat, they're going immediately to get a what? A throat swab, and they're going to get, and nobody's doing anything about it. But if you've got some teenager who all of a sudden has painful urination, God, they're sweating it out until the signs and symptoms go away, aren't they? And guess what? They're still infectious. But if they had gone, because they didn't have that shame and stigma associated with ignorance, then we wouldn't have as many of them. And there's nothing to be stigmatized about it. But there is a lot to be learned. And with HIV, there's certain even types of sex that is less likely to be transmitted through. Oral sex can be a very easy way to transmit this, not because it's in the saliva, but because it's so high in semen. And mouths has so many what? Yes. So many, there's so much vascular. That, if you just floss your teeth hard that day, but do floss your teeth, okay? So anyway, but you know what I'm saying. Where vaginal intercourse is less likely to transmit than anal intercourse. And so this is why we do see, it's much more likely in women who don't have foreplay. Women who don't get foreplay have a much more likely chance of having transmission of HIV virus. Why? No lubrication, which is going to cause these micro abrasions and whatever. So that's what we found out when we actually went and looked at areas where it's so prevalent in heterosexual sex. In some areas, it's so prevalent. And when epidemiologists and anthropologists went in to look at the culture of how these groups of women were getting it so much easier than other areas, the one difference was that in that culture, women weren't getting foreplay. That was the difference. That's sad on so many accounts. So, no, just saying. But right? No, it's nothing funny about this, right? But that's right? So HIV is not easily transmitted. It's really not. Can it be transmitted from mother to child in utero? Yes. But with treatment, the risk of that goes way down. This is why planned pregnancies in 2019, I don't even understand. Of course, planned pregnancies, so that you can get early detection, know, and take treatment so that there's less of a risk. It's really high in numbers in breast milk. So can women who are HIV positive breastfeed? No. So we know that, so making sure. I remember, guys, I was part of this this whole thing in the 80s, where we were going in and whole communities in certain cultures in Africa were being just decimated, right, by AIDS, because they had gone from HIV positive to full-blown AIDS. So women, children, men, young, old, dying, right? So developed countries went in and tried to start sending in medications, and we're sending in medications and sending in condoms and trying to do cultural changes with just allow education, just trying to let people know how it spread and whatnot. But it was so sad that was happening. And we were preventing mothers from transmitting it to their fetuses. But then guess how the babies were getting it? Through the breast milk, because they didn't have funds to get formula. So they were still having to feed their babies. And that was tragic, because that went on way too long after we knew that was the case. But that's the only way you can feed your baby. That's the only way you can feed your baby. So anyway, all right, so I got a little bit off track, not really. So how do y'all feel about this? Let's take a quick break. We're going to come back and do, we're going to come back and y'all take a break. How about 10 minutes? 10 minutes? We'll be done by 1, okay? So 10 minutes.
Are we doing all after that?
No, we're doing that whole thing that they're making me do. That means, and you will be getting credit, a lab quiz, participation for that. Anyway, so we'll do that in just a bit. But let me go over these antibody classes again. Again, antibodies are what? What kind of molecules are they? Proteins. They're proteins, and they're made in response to antigens, specific foreign antigens. There are a few naturally occurring antibodies we talked about, and that was in the what typing? ADO typing. But it's rare that we have naturally occurring antibodies. These are we usually think about antibodies in response to foreign antigens that we get exposed to. So guys, I am going to want you to, and you'll see, and of course it's going to be open book, but I can't express to you how you should really understand that there's different kinds of antibodies. And it's based on their organic structure. Now, of course, you're not going to have to draw antibodies. I would want you to. We already know that structure dictates what? Function. Exactly. So when these antibody classes that we're talking about, the reason I want you to know about them is that because we know when they are produced, we can use them for diagnostic purposes. Everybody hear that? Because that was the important part of this. The first time you get exposed to something that's foreign that you've never seen before, this is the class, IgM antibodies, that will start to rise. Do they start to rise immediately, or does it take them a while before you learn the antigenic structure and you figure out how to make these? It takes a while, doesn't it? This is why you're having signs and symptoms of the disease. But as soon as the titers of IgM go up, what are happening to your signs and symptoms, hopefully? They're going away. So I would want you to know that. So if somebody has been sick for at least, and it takes a while, at least a week or so, of certain signs and symptoms, and you're thinking to yourself as the practitioner, it might be this disease, and we have the ability to test for it, what titers are you going to ask for? IgM. Exactly. Okay? Now, I told you, though, that when you are first exposed to a disease and you get these first initial antibodies, they disease, but then you would have these memory B cells. And so the next time you get exposure, what kind of class is it going to be? IgG. And these IgG titers are the predominant titers, because these are the ones that you have to everything you've ever had memory for. These predominant, and these are also the class that can cross the placenta. That's a very good thing, because if Andrea and Julie are both pregnant, right? They're both pregnant, and that neither one of them have had immunization for measles or had the measles. So the measles virus is completely foreign to them. It's their system. And I come in hocking with it, or sneezing. I don't have to be this close. I can be 15, 20 feet away. And she's like, so I can be 15 feet away, and guess what? It is easily transmitted. They both get measles. Now wait a minute, let me change that. You had never been exposed to it, but you had an immunization. So your immunization gave you protection. Your antibodies crossed and protected your fetus from hers on, because she doesn't have it. So she gets measles. It is, just like probability, it's likely she's going to get okay. She's going to be okay, but what's it going to do to her fetus? can devastate the fetus. Right.
That's a foreign, it's two foreign bodies, or, I mean, what causes the measles to attack the fetus?
The measles itself virus can cross the placenta. Some can cross the placenta.
So just not having the protection at all.
So where she wouldn't even get it, because as soon as the measles virus is in her system, her own antibodies are. It's never going to be able to get to the fetus. Her fetus is protected. You get what I'm saying? But also her antibodies are crossing, protecting, even if it did get the fetus. So let's say she has, so she has antibodies that's crossed the placenta. She gets exposed to fetus. She gets exposed to measles. It's probably never going to get to the fetus. But even if it did, the antibodies are protected in that fetus. She has the baby, and hopefully doesn't have it in Walmart the week after she has it. But if she does carry it to Walmart and there's measles at Walmart, your baby is still protected for a few weeks because those antibodies cross and are in your baby for a few weeks. So you're still protected. The baby is still protected on you.
Well, would
You, on the other hand, are not.
would encourage the baby to get just the shot, the immunization for it, or
The babies get the immunization at a certain prescribed time during pediatric development. So the baby's not going to be protected. Her baby would be protected, yours would not. But hers would be protected for a few weeks after delivery because her antibodies cross the placenta. So another just good reason for people to get immunization. What'd you say?
I was just gonna ask if she were to breastfeed, would the baby have more antibodies on?
That's a different kind of antibodies. These are IgA. And a lot of those antibodies that are found in IgA, which is like breast milk, saliva, and all of that, they're very protective too. But oftentimes they're protective of the GI. They're actually coating the GI epithelial tissues. And babies are most at risk for dehydration. So this is why they encourage women to, even if you don't want to breastfeed, and I'm not one of those crazy people who say, you should be breastfeeding, you should be ashamed of yourself if you're not, because that's not really true. People should breastfeed if they want to. But a reason to breastfeed is that first colostrum, that first milk, is so rich with these mother's antibodies that are coating that neonate's intestinal tract. And they're the most at risk for losing fluids because they're not protected this way, right? They're much higher risk of losing fluids that way. And especially if there's GI bugs, if that makes sense. Okay, so IgA is going to be found in intestinal secretions, saliva, tears, milk. That's where these are going to be found. IgE are going to be increasing. The titers of these are going to increase when there are hypersensitivities and or parasites, worms and whatnot. These IgE, I know they sound like they're real pain, and they kind of are when these titers are high, because usually people are going to be exhibiting symptoms of hypersensitivity reactions. Not usually, they kind of are. So when there's too much of this, sometimes they try to give people allergy shots to decrease the amounts of these and get somebody to seroconvert to this. So the first IgM is on initial exposure. IgG is the predominating one that you get when the memory cells are going to do, memory B cells. IgE are in response to allergens or parasites. Maybe you can remember this one because E and eosinophils. Don't you know these are the white cells that are in response to those two? And then IgA are in body secretions. IgD I do not ask you about because it's actually, I told you that the inactive cells become active, and this plays a role in kind of that. So I don't really care that, I don't think you'll ever be asked that or get a titer for it. Anyway, so how do you feel about those? You feel okay? This is that big old monocyte, that's phagocytizing this pathogen here, this foreign body. Through the lysosomes, they're actually digesting it. And this was what I meant about spitting out some of it, but also presenting the antigen on its surface, antigen presenting cells. She was the foreign body, and I took her in, and then I presented the antigen out there so that the other cells could become activated. Interleukins are between white cells. These are those messengers between white cells. These are just some of the groups. I'm not going to ask about them, but we know they send out like siren-like alarms that pull these other cells, making them become active. So these are these molecules. And they really are hormone-like messengers, because don't hormones elicit a response in another cell? Don't they do that? So I mostly just want you to have heard of these. So lymphokines, obviously produced by lymphocytes, monokines by monocytes that become macrophages. I'm not going to ask you about them. I just want you to know there's this whole group of them. And then you can see pictures of how they end up bringing things into the cell, whatever. I don't ask about that either. Now, how they attack, antibodies attack. Sometimes the antibodies actually through something called agglutination. Y'all saw this. Did you see on your little cards when the cells started clumping, the antibodies were hooking up? They were hooking up with specific antigens on the cell membrane, and y'all saw that agglutination, it was called. That agglutination can cause precipitation out. If this were not a red blood cell, it does that too. But if this were not a red blood cell, if it were a pathogen, and it had locked them all up, the pathogen can't invade your cells when that happens. It's not going to be able to invade. And so then it can precipitate out those particular pathogens. That's just one of the ways that they can actually work. And so anyway, it just gives you that idea. This is, I think, a very amazing slide, because you see on first exposure, and you get introduced to the foreign antigen, whatever the disease might be. And did we say all of them are going to give you signs and symptoms? No. But the ones that may give you signs and symptoms. The first few days you don't even have detectable levels, but after a few days, and you're feeling a little bit sick here, right? But then you start building the IgM. The antibodies are doing their work. Now, at the same time, what is also happening? These were the Air Force. These were the bombs. But what else is happening? Don't call them Marines on the test, but what are they really? They're the CD8 cells, the cytotoxic T cells, right? They're also doing cell to cell. And then what's happening to your signs and symptoms? They're going away, right? And then the titers come down a little bit. On second, third, fourth, dozen exposure, does it take long for you to get a titer? Look at this IgG. It just goes sky high on exposure, right? So, and it does it within minutes, actually. So I think that's actually a beautiful thing. There are all kinds of pictures where you see the helper cells initiating this entire thing, and why that is so important. Y'all Google, if you have a chance to Google an animation of an actual T cell destroying the cancer cell. You know, these little T cells might be smaller than some of these cancer cells, but they are no joke. They are those, you know, I talked about the Marines. They're no joke, right? And they can just destroy those cells in a quick hurry. We have cancer, we have cells that are losing control mechanisms every day. But that's okay, because we have strong immune systems, meaning we have strong numbers of those cells that are recognizing cells that have stepped out of line and killing them, right? That's what's happening right here. They actually, a little illustration of this is that they're doing this cell-to-cell combat, perforating, which is kind of like stabbing those cells, and they get rid of them. So suppressor T cells. I wish I could think of the, I wish I could think of the other word. Kristen, in your micro book, the micro text is actually called suppressor T's a different name. And that's how much of an impression it made on me. I can't think of the other name. But it's a word that sort of means suppressor or kept in check. So anyway, but it's another name. And so we know these play a role in preventing autoimmune diseases. So people who have autoimmune diseases, maybe their suppressor T's before what's that called, right? Memory, and we have memory. Now I want to give you two definitions, and then we'll be done for this for now. And then that's, I think, enough for today. And I'm going to give you all this little survey. And then y'all can go. Now look, guys, I want you to, you're going to be asked about active versus passive immunity. Don't make this harder than it is, all right? Look, so active immunity means that you're making your own response. Active means that you're making your own antibodies, you're eliciting your own cytotoxic T cells, and you're doing it yourself. And one way to have that active immunity is to actually be exposed to something. Won't you have active immunity if you get exposed to something the first time? So that's called natural active. Because in a natural world, aren't we exposed to a lot of foreign things? That's natural active immunity. You're making your own response. You're protecting yourself. Now you can have artificial active where you make your own response, but it's because you got an immunization. You got a vaccine, an immunization. Fair enough? And then you still built your own response. That's active, you being active. You can also be passive and get immunity. You can be passive as a baby suckling at your mom's breast. And as a baby suckling at the breast, what's the breast milk giving? IgA antibodies. So you can be given antibodies through your mother. You can get them through in utero, right? You can get them in utero, crossing the placenta, protecting, the fetus is being protected. The fetus isn't doing anything, it's just getting protected by the mom. The fetus is breastfeeding, getting protection from the mom, right? Or you can actually, as an adult, and you are working with a patient who's got hepatitis B, and you already know that you've got titers to B, you're kind of protected against B, but you have some sort of, I don't know, accident, maybe they're bleeding all over you because you're a first responder, and you know, you wear gloves, you got all the stuff you're supposed to be doing, but blood is spewing, right, and other body fluids. So if you find out that person was active for hepatitis B, you have antibodies, but if you go, you should also be supposed to go because they're going to give you shots of antibodies that are already made, gamma globulin that's already made, so it's going to boost your system. Even though you already have it, they're going to also give you shots. So passive can be from a mother in utero or breast milk, but passive can also be given in shots. So does that make sense? Two different types of protection. Active, you're making your own, either naturally or artificially, and passive that you get naturally from your mom or artificially through immunoglobulin shots. How do y'all feel about this? This lymphatic system, is that kind of new information to you guys? A lot of students tell me that this is the system that they know the least about coming in. But still, even though they know the least about it, still they don't think it's as hard as the endocrine or the cardiology, cardiovascular. But I hope you felt like it made sense. You feel pretty good?
Can you get to gamma globulins even if you're not immunized, or is it only good? Like, because you were using Julie like a Hep B